Well, welcome back. Now we can talk about the process of translation. The last lecture left us off right at the point where we were getting to translation. We've already previously covered DNA replication during S phase of interphase. And then we have transcription, which occurs in the eukaryotic nucleus. And then we have RNA processing, which occurs in the eukaryotic nucleus. Then we have nuclear export of that mature mRNA transcript. And now we're finally up to the point at which we use that mature mRNA transcript in the cytoplasm, and we translate it from the language of nucleic acids, because it's composed of RNA, to the language of amino acids, which is going to be the language of proteins. So before we get into it, let me just say a few things about proteins. Protein structure and function are absolutely linked. Proteins perform an extremely wide variety of functions in living organisms, which is why we talked earlier about how when they were debating the chemical component of heredity, proteins was the last challenger to DNA, because proteins do so many different things. So you could see that proteins can be responsible for enzymatic reactions that can generate light or catalyze chemical reactions. We have fibroin, which is spider silk protein. This is a very, very high tensile strength protein, and so it's structural in nature. You have ricin, which is a protein poison. So they carry out a number of different functions. Protein functions vary widely, which is what it says here. Proteins perform a wide variety of functions in living organisms, and all 20 amino acids 20 naturally occurring amino acids that you find in these proteins have some characteristics about them. So there are three characteristic groups that they have. They have an amino group, which is going to be an NH3 plus in most situations, not necessarily an amine, which is NH2. They've got a carboxyl group, which is usually going to be a COO minus. Just imagine if we were in neutral solution. This is an acid, this is a base. And so they're tethered to one another. So they're going to react. And so you're going to basically the acid's going to donate a proton, it's going to end up over here, the base is going to accept a proton, so we have an NH3 plus and a COO minus. That's important because we're going to see that proteins start at their amino end, their N terminus, and end at their C terminus. Now this is just a schematic for one amino acid. Once we have a chain of these connected, then we'll have an N terminus and a C terminus, and that's basically like 5' to 3' in DNA or RNA. The starting end is going to be the amino side, the N terminus, and then the terminal end, the end of it, is going to be the carboxy terminus, the C terminus. But in addition to an amino group and a carboxyl group, we also have the all-important side chain. So you could say they have a unique side chain, which is symbolized by an R. That's just organic chemistry nomenclature, right? So R is your variable in organic chemistry. R usually represents a carbon group. And so what you have are a whole lot of different versions of R. The 20 naturally occurring amino acids, their R groups are shown in blue. And so they could be something as simple as a glycine, which just has a little hydrogen and nothing else, to something as complex as, say, tryptophan. And so tryptophan has, as you can see, it's got a heterocyclic aromatic amine as its side chain. Nothing you have to, I'm not going to ask you that, that's just organic chemistry speak. But basically it has a complex structure for its side chain. So amino acids are going to be joined together by, for those of you that have taken organic chemistry, you're going to form an amide bond, but that amide bond in biochemistry is called a peptide bond between amino acids. So you get C double bond O connected to an NH. So we're going to pull out the blue, which is a water. And so it's a condensation reaction or a dehydration reaction. And so by removing a water molecule, you can put these two things together, and now you've created a dipeptide. So these are just general amino acids, right? We don't know what amino acids. This is the side chain 1, side chain 2. And so here you can see, we pull the water out, connect them. And so we have a peptide bond. And that peptide bond is important because there are different levels of structure to proteins. And the peptide bond, so connecting amino acids together, that first just string of amino acids that you have, whatever order the amino acids are that you put together, that is what's called primary structure. And I'll have all this on a slide in a second. But then what's important is that there are also areas of secondary structure. And secondary structure comes from the peptide bond being able to hydrogen bond. You have a partially negative oxygen here, you have a partially positive hydrogen here. So you can imagine that, here, let's draw it out. So if you had, excuse me there. So if this chain of amino acids just kept going, right? Well, you can imagine that it might connect back around. And so if it does that, then we have the ability for another peptide bond in this region to actually form a hydrogen bond here, or here, or here, or here. And so you get the idea. Basically what happens is that that peptide bond is going to be important not just for linking the amino acids together, but for also basically providing the hydrogen bonding necessary to create the next level of structure. So polypeptides are polar. I've already said this aloud, that by convention they are read from N terminus to C terminus. That conforms with the way in which information is encoded. And so the information is encoded, and so it's trying to be expressed, and the way the protein is created is N terminus to C terminus. It's very analogous to 5' to 3', right? That's the way the information is encoded. That's the way the nucleic acid is created. Same thing here. The polypeptide is composed of amino acids, and so it reads N terminus to C terminus. The way it's constructed is N terminus to C terminus. So after a polypeptide is formed, as I said, there are multiple levels of structure. And so oftentimes these proteins need to fold into a three-dimensional shape because we started with the idea, the premise, that protein structure is absolutely critical for function. And so it folds into a three-dimensional shape determined by its amino acid sequence. So here you can see primary structure, the amino acid sequence, right? The linear sequence of amino acids in the polypeptide is the primary sequence. And that's going to govern all the other levels of sequence. Because in order to have these peptide bonds, once you have these peptide bonds, you have the possibility of forming secondary structure. Hydrogen bonding between the peptide bonds allows us to have a beta-pleated sheet, an alpha helix, which is an alpha helical structure. Random coils are not as random as they sound because they tend to hold their shape, and that's because of hydrogen bonding between those peptide bonds. Then we haven't even talked about the side chains. So the side chains are capable of interacting with one another. And so when those side chains interact with one another, they cause the polypeptide to take on a three-dimensional shape. And that three-dimensional shape, again, critical for function, and that is what's going to the side chains are going to be what is responsible for the formation of tertiary structure. Lastly, if the three-dimensional shape is just one of multiple subunits of a larger protein, then these subunits combine, and that gives you what's called quaternary structure. So the folded polypeptide, once this is all done, either here or here, you finally can call it a protein. It has to be, it's non-functional when it's just a long floppy polypeptide, but once it takes on its three-dimensional shape, then it's in its functional form, and that's when we usually call it a protein. So a protein's amino acid sequence determines its three-dimensional structure and function. There are 4 levels of protein structure. As we said, here they are in bullet points. Number 1, you have primary sequence, or primary structure, I should say. Primary structure is the linear amino acid sequence in the polypeptide chain. It's held together, primary is held together by peptide bonds. Secondary structure, as I said, is the three-dimensional configurations that result, so beta-pleated sheets, alpha helices, random coils, for example, that result from interactions between the amino acids NH and CO groups, because we have a hydrogen and we have an oxygen, so we have the ability to hydrogen bond. And then tertiary structure comes from, so remember, secondary structure is held together by the hydrogen bonding between the peptide bonds. Adjacent peptide bonds, about 3 or 4 amino acids away from each other. They're called residues once they're linked. So these are all residues, amino acid residues in a polypeptide. And so if you go about 3 or 4 residues down, then you can, you'll have enough space to actually utilize the hydrogen bonding capacity of the polypeptides and make them hydrogen bond with one another. So then we have tertiary structure. Tertiary structure is folding of secondary structure back in on itself. So now we can use the side chains that might be sticking out that can basically form what are called salt bridges. So that's plus and minus going together. There could be hydrophobic side chains that stick together because they're just burying themselves with one another in what's called the hydrophobic effect. There could be covalent linkage where you actually form a bond between 2 sulfur residues. So cysteine can form what's called a disulfide bridge. And what did I say? I said salt bridges, disulfide. And of course, you could also have just hydrogen bonding. And so they could still hydrogen bond as well. So you can have basically polar interactions. Lastly, the fourth level of structure for proteins is quaternary structure. And quaternary structure is when you have interactions between two or more polypeptide chains. Like, for example, this is an example of myoglobin. So it is composed of four subunits, as you can see. In the past, I've used hemoglobin as an example in your red blood cells, and that's 2 alpha chains, 2 beta chains. Again, that is made up of, it doesn't always have to be 4. Those are just 2 examples. You can have, you might recall when we were talking about wrapping up the DNA to get it to fit into the nucleus, we talked about the histone octamer. So the histone protein is made up of 8 different subunits, and so that's going to have quaternary structure, definitely, because of the 8 subunits. So let's look at secondary structure. So this is what's called a ribbon structure. This is how we represent, in biochemistry, how we represent proteins. And so we have to find the N terminus and the C terminus. One way is to use one of these beta pleated sheets as an arrow, right? So you can see, for example, here's an end. So let's follow this end and see, oh, good, we're following the arrows. The arrows are pointing in a certain direction. So those arrows, those green arrows, represent beta pleated sheets. Here's an alpha helix, and then it goes and then keeps going, beta pleated sheet, and then here's another alpha helix, and there's the C terminus. And so that would be how we would represent the N and C terminus, and then this polypeptide, once it's taken on, obviously there's primary structure here because you have a chain of polypeptides, or sorry, a chain of amino acids, I should say, forming a single polypeptide. And then this single polypeptide basically is capable of forming areas of secondary structure as indicated by these green arrows or these alpha helices. So that would be secondary structure. The fact that the secondary structure is not just all over the place, just flopped out and just basically this protein's denatured, but the fact that it's in a compact three-dimensional shape, a specific shape, indicates that we have tertiary structure, folding of secondary structure back in on itself. We don't have any quaternary structure because we only have one polypeptide here. But if we wanted to see some quaternary structure, we would need 2 or more polypeptides. So we could add another polypeptide to this. And so now we've got 2 of them. They're basically going to be linked together by interactions of side chains between these 2 alpha helices. And so now we have a dimer. And so this now is a protein that contains quaternary structure because it contains 2 or more, in this case 2, polypeptide chains. All right, so what we've been working our way up to is basically a complete understanding of the genetic code and how genes are expressed, how you take the information encoded in the DNA and utilize that information and then make RNA and then eventually make protein that can actually carry out functions in the cell. And so the information to encode a single amino acid is carried in a sequence of 3 nucleotides. And I feel like we may have talked about this before, but it's worth mentioning again. So if you have 1 nucleotide, you could only code for 4 different amino acids, and there are 20 amino acids. If you had 2 nucleotides, if you had a sequence of 2 nucleotides, one of 4 bases at each position, that gives you 4 times 4, 16 different possibilities. And so that's still, only 16 different possibilities, and so that's not enough to code for to code for 20 different amino acids. But if you have 3 nucleotides, you have one of 4 bases at the first position, 1 of 4 bases at the second position, 1 of 4 bases at the third position. That's 4 times 4 times 4, 4 cubed, 64. And that's more than enough to code for the different amino acids, the 20 different amino acids. And so the 3 nucleotides give 4 to the third different combinations to encode the 20 different amino acids. You can see them here represented in RNA. And so I may have said before that a triplet in the DNA is called a codon in the RNA. And so, basically, let's see. A, U, G is our start codon. So that is methionine. And so it codes for methionine. So you can see AUG, it's got a little asterisk. Oh, that's nice. Look at that. The laser pointer is perfect. Oh, I lost it. It was perfectly on the asterisk. Oh, I can't get it back. Oh, there we go. Got it. All right. So anyway, AUG also serves as the principal initiation codon. You have no idea how many sidetracks and things like that I have to edit out of these lecture videos because, I don't know, apparently I have a low attention span. But basically, each triplet is called a codon. And so we know that there are 64 codons. They, we only have 20 amino acids, and so as we've said before, there's a lot of redundancy in the code. You can see like there's a lot of codons for leucine, CC anything is proline. You can see there's quite a few arginines, all right? And so basically the code is degenerate. What that means is that that third position is not as important. It's called the wobble position. A second ago, I just told you CC anything codes for proline, right? Well, here's AGG, which doesn't match up perfectly with UCU in the mRNA. This is the mRNA, so this is the codon. The tRNA, remember the tail end of the last lecture, I'd mentioned what an anticodon is. It's the part of the tRNA that reads. It's the tRNA is the translator molecule, right? It stands for transfer RNA, but it speaks both languages. So it's bringing in a serine because it has a anticodon AGG, and UCC on that previous image codes for serine. And so if you had a UCU, you could still get a serine in there because the first two match up, and 2 out of 3 is good enough. That's why it's called a degenerate code. That's why that third position is called the wobble position. This literally is going to wobble when it's in there in the ribosome in the translational machinery, but it's going to be immobilized long enough for the serine to be catalytically added to the growing polypeptide chain. So we're back to this. So serine was, which one was it? You know what? I forgot. I think it was AGU, wasn't it? And so we were looking at an AGG, which isn't going to normally code for serine, but you could get a serine in there. And so then, of course, there are also stop codons. Stop codons are UAA, UAG, UGA. And I also mentioned our good old start codon. So you got to have your start codon. Okay, so how do we take advantage of this code, right? How is this code actually utilized? So the code is kind of the theory as to how this all occurs. And now what we're up to is basically how this all occurs. And so the nucleic acid code in a mature mRNA is translated, the physical process by which we're actually going to make some proteins, into amino acids to synthesize polypeptides. And so in eukaryotes, mature mRNA are going to be exported out of the nucleus, right? They have to go through that processing and everything like that. They have a cap, they have a tail, they're spliced, and they're exported out of the nucleus. And then once they're in the cytoplasm, they can be translated, and they can be translated many times over. So this is not going to get consumed. The mRNA isn't going to get used up. It's out there in the cytoplasm for as long as it can be, as long as it remains stable. And while it's out there, a ribosome is going to assemble on the N-terminus, and it's going to basically large subunit, small subunit, some other factors. They clamp down on there, and then they start reading the information. 3 by 3 by 3 by 3. They just go ticking along it. And as they get out of the way, and they're making amino acid, growing polypeptide chain, as they're moving down the RNA, the mRNA, another ribosome can clamp on and start. So a mRNA molecule is going to be studded with ribosomes many times over because it can be utilized simultaneously by multiple ribosomes. As long as that start codon becomes available, another ribosome can basically clamp down on it, start moving along it using that information. Just imagine, like, I don't know, a billboard up that like multiple people are looking at, right? It's not like what one person reading the information presented on that board is going to preclude other people from reading it. And so eventually, if you have so many people, then you won't be able to see the information on the board. And then, yeah, you wouldn't be able to have access to that start codon. Ribosome couldn't clamp down because that mRNA is saturated with ribosomes. But as soon as they're moving, and they'll move out of the way. And so then another one can clamp down on that. So here's what that looks like. So this is an RNA molecule. You can't even see it. It's in there. It's a scanning electron micrograph. There's an mRNA molecule in there, but you can't even see it because it is just studded with ribosomes. These are these large, somewhat spherical, blobs are ribosomes. And if I were to ask you which end is the N-terminus, which end is the 5' end of the mRNA, which end is the 3' end of the mRNA, the 5' end is the starting side, you would look at this, and what I'd hope you'd be able to do is look at, okay, well, this is a ribosome. It's got a pretty long thing coming off of it. That would be a polypeptide. Here's a ribosome with a really short polypeptide coming off of it. What that tells me is that this ribosome has read a lot more information and created a polypeptide accordingly that's much longer than this one. So this one just got started. So that means that this is the starting side. This is the 5' end of the mRNA molecule. There's an arrow here to indicate that, but of course, if there wasn't an arrow there, then you would have to just be able to look at this and figure it out. And as you can see, there are quite a few ribosomes on this mRNA molecule. And then on top of that, the polypeptides hanging off of them are longer as you progress down the mRNA, which makes sense because you've read more information. So one mRNA transcript can be translated many times over. It is currently being translated many times over, even simultaneously by lots of different ribosomes. Now, there are 4 stages in protein synthesis. So we're going to go through the 4 stages. I'll talk about them. I'll show you some static images. Then we'll do, of course, a video, an animation. And if the animation doesn't, present very well on your machine, then of course, just please, go to our course website. I have it posted separately there. Then we'll talk about, some of the other aspects of translation. But let's start with the process. So the very first thing that needs to happen, this isn't like one of the 4 stages. This is like before anything really happens, is because your stages are going to basically be, initiation, elongation, etc. So there are 4 stages in protein synthesis. And so we're going to start with tRNA charging. So before we even get started with translation, we need to charge our tRNAs. So remember, here's our anticodon loop. So this thing needs an amino acid attached to it. There are enzymes that do that requires energy and amino acids. You got to have your tRNAs. So there's tRNA charging. So we call this a charged tRNA once it has an amino acid on it. So it basically, if it had like tyrosine, it would be tyrosyl tRNA. If it had cysteine, it'd be cysteinyl tRNA. And so those are the charged tRNAs. Then once you have that, so here's an example of a cysteinyl tRNA that I just mentioned, right? So this is a charged tRNA. Then once you have that, you could start the 4 stages of protein synthesis. And so the first stage is, guess what? Going to be initiation. And so initiation is basically what you have here is, you have initiation factors, which we didn't talk about, but you have your small ribosomal subunit, you have your large ribosomal subunit that has to come in, you have your tRNA molecule, you have an amino acid, you have to assemble all of this. And so now, once it's fully assembled, what you have is an E site, a P site, and an A site within the ribosome. The E site is the exit site. It's for the spent tRNA. The P site is the growing polypeptide chain. And the A site is for the next amino acyl tRNA. So the very tail end of initiation is when that second tRNA with the second amino acid comes in. Once that happens, then initiation is complete. And you can form a peptide bond between these 2 amino acids. So let's take a look at that. That is called elongation. Oh, I guess one last thing. So initiation, like I said, requires factors that are called initiation factors. You'll see them. You might see a few of them in the animations, but generally they won't be included in my static images that I'm showing you. So basically, those are required to help assemble the small and large ribosomal subunits. So I just use the small and large ribosomal subunits for simplicity's sake. And then, of course, we have the mRNA template that's also necessary. But now, if we have our 2 tRNAs, we have our 2 amino acids, we have our mRNA, small and large ribosomal subunits, initiation is complete. And so then we get into, so that's why this is coming. As soon as this comes in, then initiation is over, and then immediately you're going to catalyze a peptide bond between the methionine and serine. So there are 2 things that need to happen here. So the first one is elongation. So the catalyzing the formation of a peptide bond is elongation. But then we have a problem. And the problem is, here, let me label them for you. So this is going to be, it's not shown, but there is what's called an exit site here. This is supposed to be the peptidyl site, and this is supposed to be the amino acyl site. And so if those are the sites, once we actually form that peptide bond, then so this is going to come in, and then we're going to form a peptide bond. This is going to release. And so the growing polypeptide chain is going to be right here. And so now here we have the different sites. The amino acyl site is the site for the next amino acid to come in with its tRNA, but that's occupied by the growing polypeptide chain. The growing polypeptide chain are supposed to be over here, but that's occupied by the leftover spent tRNA. The spent tRNA is supposed to be over here in the exit site so it can be jettisoned. And so we have a problem. Everything's in the wrong place. The solution is translocation. So this is the very tail end of initiation. This is elongation. And this is what I was talking about. So now we have everything in the wrong place. So here, actually, let me label this one because it's a little easier to see it. So you can see that we're supposed to have an E site here. We're supposed to have a P site here and then an A site here. And so what that means is, again, E site is the exit site. All right, you can see exit site not shown, so it'll be in the animation, but exit site not shown. And so this is a used up spent tRNA that could be jettisoned and recycled. We can stick another amino acid on it, use it again. It's not garbage, but it is in the wrong place. It's supposed to be in the PE site, sorry. It's supposed to be in the exit site because it needs to be jettisoned. The growing polypeptide chain is supposed to be in the peptidyl site. That's what that means. Peptidyl meaning the growing polypeptide chain. And so the growing polypeptide chain is supposed to be right here, but it's not. It's occupying the A site. And that's a problem because the A site is the site where the next amino acyl tRNA is supposed to come in. And so everything is in the wrong place. And the solution is step 3, which is translocation. If we just ratchet the whole ribosome and all these, these things stay where they are, but we ratchet the ribosome over 3 bases on the mRNA, that's what translocation is, then this would now occupy the E site. This would occupy the growing polypeptide chain would be in the P site. And then the A site, where this is right now, would be over here, and it would be free and open for the next tRNA, amino acyl tRNA to come in. So that's what happens. And so that's what happens. So basically translocation occurs, and you can see the movement. It's amazing, isn't it? And so everything moves over, and so now you have, basically the E site isn't really shown in these images, but this is jettisoned from the E site. This is now in the growing polypeptide chain, the P site, and now the A site is available. You can see they're labeled here, so it's available for the next amino acyl tRNA to come in. And so what happens now is that steps 2 and 3 repeat many, 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 many times over again. And so you have elongation, translocation, elongation, translocation. I worked on a very small protein when I was in graduate school. I think I may have mentioned it. It was involved in the onset of Alzheimer's and Parkinson's disease. It was casein kinase 1. It was 545 amino acids. So I would have to sit here and say, okay, initiation, elongation, translocation, elongation, translocation. I would have to basically do that 544 times. Then we can get to termination. So these steps 2 and 3, basically repeat until you get to a stop codon. And once you get to a stop codon, then I guess you could call it step 4 for termination. But realistically, it could very well be step 546 or 7, I guess. So step 4 is termination. Termination is, elongation ceases once a stop codon is reached. And so those stop codons are UAA, UAG, UGA. Release factor binds to the ribosome and the complex falls off. So basically, again, you put in an obstruction and then the translational machinery falls off. Everything's recycled. You can reuse those subunits. You can reuse the tRNAs. You can reuse anything. The only thing that you don't reuse is because you just created it, and that's the polypeptide. And now the polypeptide is made, and you can then go on to the next step, which would be like, say, for example, folding it and turning it into the proper 3-dimensional structure to make a functional protein. So like I said, mRNAs, tRNAs, everything can be used again to make a new polypeptide. Everything's recyclable. Okay, so here I have a video, and again, if you haven't seen this one already, it's posted. If this doesn't show on your machine, then it's posted on our course website. And so you want to start the molecular biology of the genes and how they work, basically translation. This video at 3:41, because you've already seen quite a bit of it in little snippets in previous lectures. So I'll start it at 3:41. Hopefully this works for you, and then we'll pick up after that. So here we go.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
Okay, so now what we have is a video of the central dogma from start to finish. So this is from, gene expression, so DNA being transcribed, then processed, then exported out of the nucleus, then Then translated, and then the protein being made. This is the whole process of transcription and translation from start to finish. Now, the link down at the bottom is for a different video. This is also posted on our course website, and it is basically just it's for kicks. It's crazy. You have to fast forward past the lecture to get to the really funny part, but at the end it's pretty hilarious, and so you might want to take a look at that. But certainly nothing I'm going to test you on. Now the central dogma video is really useful, so let's take a look at this.
The DNA double helix contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay. Now, I've said many times that protein structure is absolutely essential for function. So let me provide you some examples of what happens when it's not folded properly. Because protein misfolding, as you can see from the title of the slide, can cause all sorts of different diseases. So mutations can alter the polypeptide folding, and when they do that, that could cause things like Alzheimer's disease, and cystic fibrosis would be another example. Cystic fibrosis is very common. I think I've talked about it before. Cystic fibrosis results from defective folding of the CFTR protein. The CFTR protein, if I haven't spoken about it before, let me quickly say that we have certain cells that secrete mucus. They are necessary to protect us, line our lungs and our trachea, and other cells that secrete mucus as well, secrete things like in our pancreas and that sort of thing. And basically, the way they do that is that they send mucus out, this protein called mucin, which is very thick and viscous, and it needs to be irrigated. Water needs to dilute it so that it gets mucus of the correct viscosity. This is absolutely essential for the life of the cell and the function of that mucus to like trap particles like viruses and things and prevent and basically keep us healthy. And so, in order to bring that water in, there's this protein, the CFTR protein, that has two transmembrane spanning regions that pumps chloride ions out. And when it pumps chloride ions out, you have an area of high concentration. Remember way back, osmosis, right? So area of high concentration, the water comes in and irrigates that. Well, basically, it's drawn by that high concentration of chloride ions, but in the process, it dilutes the mucin, and so you get mucus of the correct viscosity. When you have cystic fibrosis, you have a mutation to the CFTR protein gene, and so then it's not folded properly, and it never gets to the membrane, and it doesn't do its job. And so the mucus out here gets really thick and viscous, so much so that it actually causes the cells to die and form cysts, clumps of dead cells, and so that would be why it's called cystic. And then also some tissues like the pancreas that secrete a lot of things turn very fibroid, so cystic fibrosis. So this is an example of, well, okay, we have a mechanism. When we make our polypeptide, we have to fold that into a proper protein inside the cell. If it doesn't get folded properly, well then, that happens all the time. We have quality control. It's just like, oh, made a mistake, recycle, and then we'll just do a new one. But if there's a mutation in the DNA, that means that every polypeptide coming down that assembly line is wrong and cannot be folded properly. And so every time you go and try and fold that polypeptide, it misfolds, and so then it's recycled, and so you have a loss of function. So here is just one example, delta 508, misfolded protein. So deletion at position 508, you get a misfolded protein. And when you have a misfolded protein, it's identified by the cell as defective and therefore destroyed before it ever leaves the endoplasmic reticulum, before it ever gets to the plasma membrane, so it can't do its job. And so this is how you end up with a manifestation at the protein level of a mutation at the DNA level. So what you basically get is a disease that is inherited. It's a cystic fibrosis is a genetic disease, and that's because really the net effect of the mutation is protein misfolding. So here's the thing though, genetic mutations aren't the only thing that causes improper folding of proteins. There are things out there that are far worse. So some proteins can refold and change their three-dimensional shape. Three-dimensional shape is so essential to function that if you misfold a protein, you can cause a different type of disease. And so protein refolding disorders are called prion diseases. And prion diseases are far scarier to me than any sort of virus or anything like that, because we didn't even know prions existed up until I'd say mid 90s or so. So these are relatively new. We don't know much about them, and there's really no way to fight prion diseases other than using dark age technology. So technology from the dark ages, which is literally, prions, burn it. And so that's literally what happens. I'll tell you more about that in just a second. So we don't have a way to come up with a vaccine. We don't have a way of coming up with some sort of medication or anything like that. It is basically just kill it and burn it, and that's how we stop it from spreading. So prions are proteins basically folded into an infectious conformation that is the cause of several disorders. And these disorders are basically things you may have heard of. And so what prions actually are these protein molecules. They don't have any DNA. They don't have any RNA. They're not like virus particles. Remember from when we talked about virus particles, they're made of both DNA and protein or RNA and protein. These are just protein. And so you may have heard of mad cow disease. Mad cow disease is by far the most famous prion disease. Mad cow disease is also called BSE, which is called bovine spongiform encephalopathy. That's the proper name for it. And then if you were to consume mad cow tainted beef, then you are at risk of developing the human variant, which is Creutzfeldt-Jakob's disease. That's a human variant of mad cow disease. So what you're looking at are, what is mad cow disease, right? So here's what you're looking at. You're looking at a cross-section of healthy brain tissue from a cow. And you can see it's a darker color, a darker purple. These are stained with the same stain. So the darker, you can see this is like a dark purple. There's more tissue, there's more matter, it absorbs more stain, and therefore it appears darker. Over here, there's less tissue. It absorbs less stain, and therefore it is not as dark. And then you can actually see these voids. That's why it's called bovine spongiform encephalopathy. Your brain matter is supposed to be dense. It is not supposed to be, it's not supposed to have holes that you could literally see through. That's on the microscope slide. Light is passing directly through this tissue, and it shouldn't be. And so what happens is these prions are what are called rogue chaperone proteins. Chaperone proteins are proteins that normally help the polypeptide fold into the proper three-dimensional configuration. And you know what a chaperone is, right? A chaperone is like imagine you were asked to chaperone like a junior high school dance, right? You know what your job is, basically make sure not too much stuff happens, that sort of thing, right? But if you showed up to that dance with like a keg and crack, you would be a rogue chaperone. You would not be a good chaperone by any measure. There is no talking your way out of that. No, that is not what you're supposed to be there for. And so that's what a prion is. A prion is a rogue chaperone. Normally, it's supposed to oversee the folding of polypeptides into their proper function, three-dimensional shape, so that they can function properly. However, a rogue chaperone, an infectious chaperone, a prion, will refold these proteins into three-dimensional configurations that are either going to cause the protein to be destroyed and degraded, or turn it into another prion protein. So that's how they can self-replicate. And so what happens is the vast majority of the proteins are going to get refolded into a non-functional form. They're going to get destroyed. That cell, so imagine this happened right in the center of this void, right? So that cell eventually is going to lose the function of that protein and all the other. This is catalytic, right? So these chaperones just refold. And so you lose a lot of functions of a lot of proteins, and then eventually the cell dies. Well, along the way, some of the prion protein had refolded polypeptides into other prions. So there are still prions in this cell. And so when this cell dies, it lyses, it spills its guts, it basically spills out some prion proteins to the surrounding cells. They get infected. And so then they basically go through the same process and then spill. And so you can see that what starts as a very small void, so if you look closely, you can see right down here is a nice small void, right? You can see it right there. And then eventually it's going to just continuously grow to a much larger void. And so what you're doing is you're destroying brain tissue. And remember, brain tissue is made up of neurons, and neurons are like wires, and they need to be insulated and they need to be connected properly into the correct circuits. And what you're doing is you're just, take a circuit board, well, don't, but if you took a circuit board, right, out of like, a desktop or something, and you just took a hole puncher and you started punching holes in the circuit board, you might guess that it's not going to function properly, right? And that's what happens in the case of a mad cow disease infected animal. They lose their ability to basically do anything. So they lose motor function. They lose their ability to even move or any, it's just really, if you YouTube a mad cow, you'll see that it's pretty heartbreaking. The animals are in horrible shape. And the fact that it's a snowball effect means that by the time you actually show symptoms, it's too late. And so there's no way to reverse this. It's not like there's a medication that will specifically go and find this prion protein, because you don't know what the next prion protein is going to be. It's rapidly changing because it's just misfolding other proteins into prion proteins. And so realistically, in the case of cattle, so dairy farmers and cattle ranchers, they can have insurance against this. When one cow is detected to have mad cow, then you basically the term that's used is you sacrifice the animal. Basically, you kill the animal, and all of the members of its herd, any animal that it had contact with, that it could have transferred the prion protein to. So remember that it's a protein, so it can be transferred via saliva. You don't have to eat one another. It could just feeding from the same trough, drinking from the same water source, can infect other cows. And because it is so imperative that we stop the spread of this before it impacts the, like for example, imagine if the beef industry was hit by one of these. Don't be naive enough to think, oh well, we don't get hamburgers for a little while. It's going to be an economic nightmare because of all the meat processors and all the equipment, all the farm equipment, all the ancillary, just even think truckers, refrigeration, just all of the different aspects of the economy that would take a hit. Mad cow would be horribly bad for our economy. And so the CDC will descend on that area once it's detected and basically take the nuclear option and essentially kill all those animals and then incinerate them, because that's the only way you will have denatured all of that protein so that it cannot infect any other animals. So it's not just bury them or something like that. It's incinerate them to ash so that the protein is completely denatured and gone. And you may not have heard. I mean, so in the 90s there was a pretty bad mad cow outbreak in Europe, and I remember I was traveling through London Heathrow and on my way to Nairobi, and I remember my friends were joking. They're like, "Well, don't eat any hamburgers when you're there, right?" And I mean that was a joke, but it was true. I mean, people would specifically not eat beef at that time in Europe or at least in the United Kingdom because of the mad cow outbreak. And we've learned from that mad cow outbreak a lot, like how it manifests itself in human beings, how long it takes to show symptoms and things like that. But if that were to happen here in the United States, it would be absolutely devastating. So my question to you is, has there ever been a mad cow outbreak here in the United States? The answer is yes, absolutely. But many of you might not be aware of that. In fact, one of them was just a little bit north of our campus, I believe in Hanford. And so basically, they happen. And then there is such an immense response that they are contained as quickly as possible, so that the beef industry and the public in general do not panic and then either stop buying beef, or you basically take a tremendous hit to the economy. So in the human variant, I'll show you some data on that in just a second. But in the human variant, so the human variant is Creutzfeldt-Jakob's disease. Prions cause normal proteins in the body to refold into new infectious three-dimensional shapes that kill cells in the brain and the nervous system. Now, if it were to happen in your muscle tissue or whatever, you could actually regenerate those cells theoretically, right? But what we don't regenerate is our neurons, right? For the longest time, we thought we didn't make them ever. We now know that we can regenerate them to a certain extent, but nowhere near the rate at which they're getting destroyed. And so that's why it affects the brain and nervous system. So before we look at the data, here's what it looks like. Here's a protein, normal, right? And then a prion gets a hold of it, refolds it into death, right? That's what you're looking at here, the refolded infectious form. And so it's quite a bit different. Even if you don't know how to look at these ribbon structures, you can see that these are certainly two different ones, right? Well, it's the same polypeptide, but one is the normal functioning form, the other is an infectious prion form. So I mentioned mad cow disease cases. So this is from 2012. That was in Hanford, I believe. Here we have a number of different cases being shown from the CDC website. You could see we have Florida in 2018, Alabama. And remember, I made this slide actually. I haven't looked recently, so I made this slide in 2019. And so, I have to check to see if there are any other, or you can check for other cases of mad cow. Now, this is mad cow specifically, right? So you can see all across the country, right? So Washington State, Texas, Alabama, California, Alabama, Florida. And so we have multiple cases of mad cow outbreaks, and they're handled by the CDC. But does that mean that people have been infected? And in fact, it's supposedly very rare, right? So the idea of Creutzfeldt-Jakob's disease being, if you were to contract it from eating mad cow tainted beef, is extremely rare in this country, and that's true. However, there are quite a few organisms that are just vectors, meaning they're perfectly fine carrying it around, just all over the place, and you should avoid them and you should not eat them. For example, can you think of any animals that might be just bopping around campus, on our campus, that are vectors for basically prion diseases? The answer is yes, right? So when I ask this question in class, there's usually some smartass that calls out somebody or some place or some, eating establishment at this point. But realistically, so most people actually come up with raccoons. They think of raccoons, but actually the most disgusting little creatures on our campus in terms of disease are squirrels. Yeah, those things that you think are cute little squirrels, no, they're not. Stay away, right? And so they are the last bastion of bubonic plague. I kid you not. You can look it up. They carry plague. They carry, of course, yeah, sure. Whatever you said they carry, yes, you are correct. So, maybe they carry rabies. I'm not sure, but why not? Hantavirus. They carry everything you can imagine, including they can carry prion diseases, such as basically what we call bovine spongiform encephalopathy when it's in cows, or Creutzfeldt-Jakob when it's in humans. So, for example, here's a story about a man who died from an extremely rare which it's not that rare. Extremely rare disease after eating squirrel brains. So, in many parts of the country, people hunt squirrels. I went to graduate school in the South, and I was invited to go on squirrel hunts, and I chose not to. Not my thing. But there are many people that hunt squirrel and eat squirrel, and so this is a man from upstate New York, and so it's not like a Southern thing. I'm not calling out the South or anything like that. So this is a man from upstate New York. He developed an extremely rare and fatal brain disorder after he ate squirrel brains, according to a news report. So he basically was brought. So in 2015, he was brought to the hospital, experiencing a decline in his thinking abilities, losing touch with reality, etc., etc. Right, and then he died in 2018 because, as we talked about, it is a slow decline. It's just a snowball effect. So he was brought to the hospital in 2015 because he was starting to show symptoms. There is no treatment. There is nothing they can do for him. He was infected with Creutzfeldt-Jakob's disease, and so he was basically just degenerating. His neurons were degenerating because of the continued, spreading of that prion or prions in his neural tissue. And so this is tragic, don't get me wrong, but I mock the headline for it being an extremely rare disease because it's not. It's just extremely rare in the sense that, it's avoidable. You don't have to eat squirrels, but it's not as rare as everyone makes it out to be. Here's another example. So a Tennessee man diagnosed with human. So this is again in December of 2018. So remember, I made these slides in early 2019, and I haven't updated them since. I will certainly do so. But basically, in my limited research, just a few months before October, now we have December 2019 in Tennessee, right? So a Tennessee man diagnosed with human form of mad cow disease. What you should know about Creutzfeldt-Jakob's disease. And so you could see that that's supposedly an extremely rare disease, right? But then here's a second case of it in Tennessee, reported in February of 2019. So and you can see rare disease. How rare is it? It is out there. It is easy to contract should you go looking for it. Fortunately, most people don't go looking for it. But if you wanted to come up with a plot to a movie, if are there any budding screenwriters out there, then forget all these movies that try and make it scary that there's some sort of disease that's a virus that is everybody always uses a virus that's spreading or a virus, and which considering the global pandemic, the COVID-19 SARS-CoV-2 virus, yeah, okay, I could understand that, but there's still a strategy to prevent that, right? Like we actually have the ability to possibly create a vaccine or something to fight that. Right now, we have nothing to fight prion diseases other than to, let the man in the hospital just, unfortunately degrade further until he dies, or, let cattle, just incinerate cattle or whatever it may be that's infected in terms of livestock. So if you wanted to come up with a really scary premise, for scientists at least, a really scary premise for a movie, make prions the thing that they're fighting, because prions are far scarier than viruses or bacteria at this point, at least to someone that might be educated about viruses, bacteria, and prions. Okay, well, that covers protein function.